Welcome to the PubMed Daily Review for March 14th. Today we scanned 2,491 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Exploring Mechanisms and Optimizing Processes. This trend explores fundamental mechanisms to optimize diverse processes and enhance system performance across various domains. This cluster covers 429 papers. Representative work includes DS-MAMBA, depth-wise separable MAMBA for hyperspectral image classification, an improved Newman fast division algorithm based on multifactor correlation for dynamic traffic subregion control, and study on the formation mechanism of color and flavor during the processing of polygonatum certainema Hua, PCH. Trend 2 of 5, Patient-centered care and health interventions. This trend unifies research on understanding patient experiences and developing effective, patient-centered health interventions to improve care and outcomes. This cluster covers 578 papers. Representative work includes Quality of Life Among People with Tuberculosis in the Eastern Cape, South Africa, a qualitative study, a telehealth-adapted dementia caregiver skills training intervention, telecare single-arm pre-post intervention study, and patient-perceived factors influencing physical activity sensor use in stroke prevention and rehabilitation, systematic review of qualitative studies using thematic synthesis. Trend 3 of 5, Disease Ecology and Global Health Strategies. This trend explores disease ecology, examining host pathogen interactions and environmental influences to develop global health strategies for prevention and management. This cluster covers 306 papers. Representative work includes Strengthening Healthcare Systems to Prevent Antimicrobial Resistance in LMICs, It Is Time to Act. An Fc-silent OSPA monoclonal antibody passively protects mice from tick and intradermal Borrelia burgdorferi challenge, and Cosmopolitan No More. Phylogenetics and reproductive mode reveal a global species complex in a marine mollusk, pteridinity. Trend 4 of 5, Cancer Prevention, Treatment, and Epidemiology. This trend integrates research on cancer epidemiology, prevention, and treatment, spanning molecular mechanisms to population-level interventions. This cluster covers 524 papers. Representative work includes populational analysis of the mortality-to-incidence ratio across 20 cancer groupings in the Russian Federation, cure rate estimation with insufficient follow-up, a median-based bootstrap correction approach, and preoperative expiratory muscle training for swallowing function in patients with esophageal cancer undergoing esophagectomy, a randomized controlled phase 2 trial protocol. Trend 5 of 5, Exploring Complex System Properties and Mechanisms. This trend explores the inherent properties and underlying mechanisms governing various complex biological, medical, and technological systems. This cluster covers 654 papers. Representative work includes alteration of functional connectivity and network properties after stereoelectroencephalography-guided radiofrequency thermocoagulation. Integrative transcriptomics unravels the central role of fatty acid elongation in early cotton fiber development, and molecular and immunological heterogeneity of eosinophilic esophagitis, insights and subtyping. Up next, our first paper out of 5, Pan-KRAS inhibition. Unlocking Broad-Spectrum Targeted Therapy for KRAS-Mutant Cancers by Huan Feng and Xiufeng Peng from Shanghai Key Laboratory of Regulatory Biology and School of Life Sciences, East China Normal University, Shanghai 200241, China. Let's break down its key contributions. Today, we're diving into a critical challenge in oncology, targeting KRAS mutations. KRAS is a notorious proto-oncogene, frequently mutated in some of the toughest cancers, including pancreatic, colorectal, and lung cancers. These mutations essentially switch on KRAS permanently, driving uncontrolled cell growth and making these tumors incredibly difficult to treat. While we've seen some breakthroughs, a significant hurdle remains: developing therapies that can tackle the broad spectrum of KRAS mutations. This paper explores the fundamental science behind KRAS-driven cancers. It delves into the molecular mechanisms, specifically how KRAS mutations, primarily at codons 12, 13, and 61, keep the protein in an active state, constantly signaling to downstream pathways like RAF and PI3K. The study analyzes the limitations of existing allele-specific inhibitors, such as those targeting KRAS G12C, and proposes the critical need for developing Pan-KRAS inhibition strategies to address the broader landscape of KRAS-mutant cancers. The paper highlights several key facts. First, KRAS mutations are highly prevalent across major cancers like pancreatic, colorectal, and non-small cell lung cancer, driving tumor initiation and progression. A groundbreaking advance was the discovery of cysteine-specific targeting for the KRAS G12C mutation, which led to the regulatory approval of allele-specific inhibitors like sotorasib and adagrasib. However, a crucial takeaway is that these current therapies are allele-specific, meaning they only target a particular mutation, leaving many other KRAS-driven cancers without effective targeted options. So, what's the big picture here? This paper underscores that while the development of KRAS G12C inhibitors was a monumental step, it's just the beginning. The real clinical significance lies in the urgent need for Pan-KRAS inhibition. Current therapies are limited by their allele specificity, meaning many patients with other KRAS mutations still lack targeted options. This work strongly advocates for future research to pivot towards developing innovative strategies that can broadly inhibit all oncogenic KRAS variants, truly unlocking broad-spectrum targeted therapy and transforming outcomes for a much wider patient population. Up next, our second paper out of 5, Dual-antigen recognition iPSC-derived CAR-T cells for B-cell malignancies. Establishment of a COVID-19 vaccine synergy strategy by Norihide Izumi and Miki Ando from Department of Hematology, Juntendo University School of Medicine, Tokyo, Japan. Let's break down its key contributions. CAR-T cell therapy has revolutionized treatment for B-cell malignancies, but we still face challenges with how long these cells last and how well they continue to fight cancer. This study explores an exciting new approach, combining iPSC technology with a vaccine synergy strategy. The core question is whether we can engineer CAR-T cells to be more persistent and effective by giving them a dual-antigen recognition system, leveraging common vaccines like the COVID-19 shot. To tackle this, researchers generated rejuvenated T cells from induced pluripotent stem cells, or iPSCs. They then engineered these cells to recognize two targets, CD19, a common B-cell malignancy marker, and the SARS-CoV-2 spike protein. These 1919 CourAGE-Ts were then rigorously tested in various in vitro assays, including cytotoxicity tests, sequential re-challenge experiments to assess durability, and proliferation analyses, comparing them to conventional CAR-T cells. The results were quite compelling. The 1919 CourAGE-Ts showed significantly higher expression of key cytotoxic molecules like granzyme B and perforin, while also displaying lower levels of exhaustion markers, indicating a more robust and younger phenotype. This translated into enhanced cytotoxicity when both antigens were present, and importantly, these cells maintained durable tumor control in repeated challenges. Furthermore, TCR-mediated stimulation by the spike protein peptide strongly boosted their proliferative capacity. This study presents a significant step forward, demonstrating that combining iPSC rejuvenation with dual-antigen recognition can create CAR-T cells with superior anti-tumor capabilities. The concept of using a widely available vaccine, like the COVID-19 vaccine, to boost CAR-T cell persistence in patients is truly innovative. While these findings are currently in vitro, they lay a strong foundation for future research. We're excited to see how this promising strategy progresses into in vivo models and eventually, clinical trials, potentially offering a more durable and effective treatment for B-cell malignancies. Up next, our third paper out of 5, reductions in recurrence in women with early breast cancer entering clinical trials between 1990 and 2009. A pooled analysis of 155,746 women in 151 trials by R. Hills and S. Steinberg. Let's break down its key contributions. Welcome. Today, we're diving into a crucial study that addresses a long-standing challenge in breast cancer, distant recurrence. For women with ER-positive early breast cancer, recurrence can be a concern for decades, but we've had less clear data for ER-negative cases. This research aims to fill that gap by examining recurrence rates and how they've changed over time for both types of breast cancer. This was a powerful pooled analysis, drawing data from an incredible 151 randomized controlled trials within the Early Breast Cancer Trialists Collaborative Group database. They looked at over 155,000 women diagnosed between 1990 and 2009, focusing on those with early-stage ER-positive or ER-negative breast cancer. The primary goal was to track the time to first distant recurrence, comparing outcomes between two diagnostic periods, the 1990s versus the 2000s, and adjusting for various treatments and patient characteristics. The results were quite encouraging. We saw similar improvements in distant recurrence rates for both ER-positive and ER-negative tumors. After adjusting for patient and tumor characteristics and improved treatments, women diagnosed after 2000 experienced a 25% lower rate of distant recurrence for ER-positive disease and a 19% lower rate for ER-negative disease compared to the 1990s. This improvement was highly significant, with a p-value of less than 0.0001. While a large portion of this improvement was explained by changes in patient profiles and better treatments, the adjusted reduction remained substantial. So, what does this mean for clinical practice? This study provides strong evidence that our efforts in breast cancer treatment are paying off, with significant reductions in distant recurrence over the past 2 decades. While much of this progress is due to better patient selection and improved adjuvant therapies, the adjusted reductions are still very meaningful. However, it's important to remember that long-term risks for ER-positive disease still exist, underscoring the ongoing need for research into even more effective and durable treatments to further reduce recurrence. Up next, our 4th paper out of 5, Phase I pilot study for hemophilia-A, AAV8 vector with prophylactic tacrolimus-glucocorticoid achieves therapeutic FVIII activity by Wei Lu and Lei Zhang from Key Laboratory of Experimental Hematology, National Clinical Research Center for Blood Diseases, Haihe Laboratory of Cell Ecosystem, Tianjin Key Laboratory of Gene Therapy for Blood Diseases, CAMS Key Laboratory of Gene Therapy for Blood Diseases, Institute of Hematology and Blood Diseases Hospital, Chinese Academy of Medical Sciences and Peking Union Medical College, Tianjin, 300020 China. Let's break down its key contributions. Good morning, everyone. Today, we're diving into a crucial area of hemophilia-A research, gene therapy. This paper explores a new AAV8-based vector, GS001, aiming to provide a lasting solution for patients with severe hemophilia-A. A key challenge in gene therapy is managing the body's immune response to the viral vector, which can hinder the therapy's success. This study specifically investigates whether prophylactic immunosuppression can enhance the efficacy of this novel gene therapy. This was a prospective Phase I pilot study involving 12 adult patients with severe hemophilia-A. Participants received the GS001 gene therapy vector at one of 2 doses, either a low dose or a high dose. Crucially, they also received prophylactic immunosuppression, either prednisone alone or a combination of prednisone and tacrolimus, starting 1 week before the gene therapy infusion. The researchers tracked key outcomes like factor VIII activity, bleeding rates, and overall safety over a median follow-up period of up to 156 weeks. The results were quite promising, especially in the high-dose cohort. At week 104, the median factor VIII activity reached an impressive 42.7 IU/dL, with all 6 participants sustaining levels above 29 IU/dL. Even the low-dose cohort showed a median of 5.6 IU/dL at week 144. Both groups experienced significant reductions in annualized bleeding rates and factor VIII consumption. While some grade 1 to 2 ALT elevations were observed, the overall safety profile was good. Importantly, the prednisone plus tacrolimus pretreatment effectively inhibited CD8 plus T-cell immune responses, suggesting a mechanism for enhanced transgene expression. This study provides compelling evidence that AAV8-based gene therapy, particularly at higher doses and with prophylactic immunosuppression, can lead to sustained therapeutic factor VIII levels in severe hemophilia-A. The finding that prednisone plus tacrolimus effectively inhibited immune responses is a critical insight, suggesting a path to improve gene therapy efficacy. While promising, this was a pilot study. Future research should focus on optimizing these immunosuppression regimens, confirming long-term durability, and assessing safety in larger patient populations to bring this innovative treatment closer to widespread clinical use. Up next, our fifth paper out of 5, clinical-grade autonomous cytopathology through whole slide edge tomography by Naonita and Keisuke Goda from Tokyo, Japan. Let's break down its key contributions. Cytopathology plays a crucial role in the early detection of cancers like cervical, lung, and bladder cancer. However, its diagnostic accuracy can vary significantly due to subjective visual interpretation by human experts. While many AI systems have been proposed to help, none have yet achieved truly autonomous, clinical-grade performance. This creates a significant gap, as existing tools still require human oversight, limiting their scalability and objectivity. This study evaluated a novel clinical-grade autonomous cytopathology pipeline. The system combines high-resolution whole-slide edge tomography with edge computing and an AI vision transformer. Researchers tested its performance on 1,124 cervical liquid-based cytology samples collected from 4 different clinical centers. The primary outcomes measured were the system's diagnostic accuracy, specifically its area under the ROC curve, for detecting various cervical lesions at both single-cell and slide levels. The results were truly impressive. At the single-cell level, the AI Vision Transformer achieved an area under the ROC curve, or AUC, exceeding 0.99 for detecting low-grade and high-grade squamous interepithelial lesions, as well as adenocarcinoma. When evaluated at the slide level across 4 centers, the system showed strong performance, with AUC values ranging from 0.86 to 0.91 for LSAL+ and 0.89 to 0.97 for HSAL+. Furthermore, LSAL counts strongly correlated with human papillomavirus positivity, and HSAL counts scaled with diagnostic severity, reinforcing the system's clinical relevance. This study marks a significant leap forward in cytopathology. By achieving fully autonomous, clinical-grade performance, this system directly addresses the long-standing issue of subjective interpretation in cancer diagnostics. Its ability to perform flow cytometry-like morphological profiling also offers a much more comprehensive understanding of cellular patterns. Ultimately, this technology provides a robust foundation for routine, scalable, and objective triage cytology, holding immense potential to revolutionize how we detect and manage early cancers.